Hi Year 10s, welcome to week 2 of Year 10 Biology. Today so last week we looked at just general biology and we talked about cells. We're now moving fully into our first unit, which is DNA and genetic information, talking about inheritance and DNA in general. So we're going to start this week with talking about what is DNA. So let's begin. So by the end of this week, there's a few things that you should be able to do, and these are things that will be able to be tested on in the upcoming test. So first of all, you need to be able to describe the role of DNA in controlling the characteristics of organisms. You're going to have to be able to use models and diagrams to represent relationships between DNA, genes, and chromosomes. Now, we will do that with some experiments. We're going to be extracting DNA from a strawberry, for example. You might even build your own DNA molecule. And you also need to be able to describe the development of the double helix model for the structure of DNA. So, those are the three things you must be able to do by the end of this unit. I'd recommend that at the end of this PowerPoint, once we've gone through all this, that you come back and you look at this and you tick off, have I been able to do these things? Because these are the national curriculum standards that you have to be able to do. So, moving on. First off, what is DNA? We're always talking about DNA. We talked about it last week when we talked about the nucleus and cells. But what is DNA? Well, for almost all things, it's basically the genetic information that contains instruction on how that organism or how that cell is going to operate, what characteristics it's going to have. DNA itself stands for deoxyribonucleic acid, and it's because of DNA that we have diversity amongst our living things. So DNA was first discovered in 1953 by James Watson and Francis Crick, and so that wasn't first discovered by them, but the model of what DNA looks like was first proposed by them. DNA has actually been discovered long before 1953, it's just we didn't know what DNA looked like and the actual double helix model until Watson and Crick came along in 1953. And we'll talk about that a little bit more when we get to the sciences human endeavour side of things. And we'll talk a little bit about the ethics behind how Watson and Crick discovered the model of DNA. The model that we have, though, because it was discovered by James Watson and Francis Crick, we refer to it as the Watson and Crick model. Now, the Watson and Crick model, it tells us that DNA is made up of many repeating units, and those units are called nucleotides. Each nucleotide is made up of 3 different components. So you have a phosphate group, which is always drawn as a circle, connected to a deoxyribose sugar down here, which is always drawn as a pentagon. And lastly, that sugar is then connected to one of 4 possible nitrogenous bases. Now, the nitrogenous base doesn't necessarily always look like that shape. I'm happy if you just draw like a rectangle at the end, and then you can put the letter of each base in there. But that is a rough image of what the shape should look like. But for now, I'm happy if you don't draw it like that. That's fine, you can just draw a rectangle. That being said, you do have to be able to draw a nucleotide, and you do have to be able to ensure that both the base and the phosphate are connected at the same points they are on this diagram, so they're always connected on the same point of the pentagon of the sugar. If you draw them incorrectly on that point, you won't get marked, or you haven't drawn the model correctly. So really important, the chemistry of DNA, phosphate's always attached to this point on the pentagon, which is actually a carbon molecule. And the base is always connected to this point, which is another carbon molecule. So that's how you draw a nucleotide. The double helix itself, though. So the nucleotides that we saw over here, this nucleotide, it repeats itself over and over again down a strand of DNA, and then you have a second strand of DNA. So here we have the phosphate group, the sugar, the base. So that there is one nucleotide. Okay? And you can see one nucleotide, one nucleotide, one nucleotide, one nucleotide, just happens to have a different base each time. So you've got thymine, guanine, cytosine, adenine, the 4 different bases you can have in DNA. And then over here, we have a second strand that runs in a different direction. So they're anti-parallel. The bases bond together via hydrogen bonds, and then the actual molecule itself starts to twist up, and it starts to look like a twisted ladder, repeatedly twisted. Basically, your phosphate and your sugar, the blue and the yellow here, they make up what's known as the sides of the ladder. So this blue line here, that is your phosphate and sugar groups of the DNA molecule. Whereas the nitrogenous bases, these parts in the middle, they make up the rungs of the ladder, so the steps of the ladder. So DNA, when we look at the actual molecule of DNA, it really looks like a twisted ladder. Phosphate and sugar make up the sides of the ladder. Nitrogenous bases make up the steps or the rungs of the ladder along there. There are 4 bases, like we talked about before. There's adenine, thymine, guanine, and cytosine. There is actually a 5th base called uracil. We'll talk about that a little bit later, though, and that's not actually found in DNA, so that's why we're not talking about it now. They have slightly different structures, and hence they'll only bond in certain ways. So, for example, adenine over here will only ever bond with thymine, and thymine will only ever bond with adenine. A way to remember that is the straight letters will bond with the straight letters. So A and T are both straight letters. They're made of straight lines. They bond together. Whereas guanine and cytosine, the same thing. Guanine will only bond with cytosine, and cytosine will only bond with guanine. And again, you can't remember that because both G and C are made up of curvy lines, so curvy with curvy. This model, that adenine will only bond with thymine, and guanine will only bond with cytosine, and vice versa, that's called complementary base pairing, and it's really important to how DNA actually works. And when we talk about DNA replication, that will make a lot more sense. For now, just know it's called complementary base pairing. A goes with T, G goes with C. Chromosomes. So chromosomes are found in the nucleus of all cells. They're very long, they're thin, linear structures. They're in eukaryotic cells, they're linear structures. And they're made up of long molecules of DNA that are wrapped around a protein called histones. In the human body, there's roughly 46 chromosomes in each of our cells, and that's 23 pairs. You get one set of chromosomes from mum, one set from dad, which makes 2 lots of each chromosome. So for example, chromosome 1, you've got a copy from mum, and you've got a copy from dad, and you get 23 chromosomes from each parent. Okay. Sex cells, on the other hand, gamete cells, they only have 23 chromosomes. So we're talking about sperm and egg. They only have half the chromosomes that a normal cell in your body has, and that's because they're combining with a gamete from another person to form a being with 46 chromosomes. There are 2 different types of autosomes. So there's autosomes and there's sex cells. Autosomes are the non-sex cells, and there's 44 of those in humans. And the remaining 2 are the sex cells. Now, that's, as you know, they're the X and the Y chromosome. They're the 2 different types of sex chromosomes. Females will have 2 X chromosomes, males will have 1 X, and they'll have 1 Y. So that's how you differentiate between male and female, is based on males will have a Y chromosome and only 1 X, whereas females will have 2 X chromosomes. Talking about those a little bit more individually, so talking about autosomes. Autosomes will be grouped into pairs. Each pair will be the same length as each other. They have a centromere in the same position. We'll draw this on the board in class. And that centromere is the point where the chromatids join. They will have a gene for a certain characteristic, and that gene will always be found on the same location on each of the chromosomes in that pair. So say we're looking at chromosome 3, you've got a copy from mum, and you've got a copy from dad. On that chromosome is the location for your hair colour. On the chromosome from mum and the chromosome from dad, the gene for hair colour will be in the exact same position. It might be slightly different. Maybe the chromosome of mum tells you you've got brown hair, the chromosome of dad tells you you've got black hair, but both of those locations will be exactly the same. So we know when we look at that chromosome, and your body knows if we're looking for hair colour, look here and you'll find that gene. So genes, I'm using that phrase a lot. What actually is a gene? Well, genes are a section of DNA found on a chromosome. Generally, there's about 2,000 genes on each chromosome. So genes are just one small section. Genes can differ by the order and the number of bases, so guanine, adenine, cytosine, thymine, that are in that DNA. The order that those bases appear is called the genetic code. And each gene will code for a specific protein. Okay, so of the 200 to 2,000 genes on each chromosome, and remember we've got 46 chromosomes, each gene codes for a specific protein. And whatever protein it creates will help express a certain characteristic. So going back to the example before, if we had a gene for brown hair, that gene for brown hair will create a specific protein, and when that protein exists in your body, it tells your hair to become or to be brown. Okay. Different genes will code for different proteins, and different proteins will have different characteristics. So, as I said before, because we get one chromosome from mum, 1 chromosome from dad for each pair, there's 2 copies of every gene that we have, and they code for slightly different variations. And we'll talk about how we determine which gene expresses itself when we get to inheritance later on this term. That's a lot of theory I've thrown at you, so we're going to look a little bit more at just the history of how DNA was discovered, more than just what is DNA. So just a little bit of a break, talking about science as a human endeavour. How was DNA discovered? We know what we need to know about DNA. Let's talk about how it was discovered. So first discovered in 1869, and this was done by isolating an unknown chemical from the nucleus of a white blood cell. The scientist that was doing this wasn't actually looking for DNA, he was looking for proteins. And when he found what we now know as DNA, he noticed this is really different in chemical composition from a protein, which is what I'm looking for. He had no idea that he'd discovered what is to be now known as DNA, or the genetic code. In 1943, so a long time after that first discovery, we've got Oswald Avery proposed that DNA was the genetic code in organisms. Unfortunately, he didn't have enough proof for this. He just proposed the idea, and that was later proven through an experiment through Hershey and Chase. And if you do biology in IB, you'll go into the experiment that Hershey and Chase did. Basically, they were using viruses to infect bacteria to see what the viruses would do to bacteria, and that would tell us whether the genetic information in humans or bacteria or viruses was either proteins or DNA. The results of that experiment clearly showed the genetic information was DNA. So that's how DNA was discovered, but even at this point that we know what DNA is the genetic code, we don't have any idea what DNA looks like. And that's where we get to the idea of the DNA model. And this is where we're talking about Watson and Crick. And there's a lot of debate about how this goes along, about did Watson and Crick follow ethical guidelines for science? Should they be credited for their work? Should other people be credited as well for the work? But it comes down to this lady over here in the picture here. It's Rosalind Franklin. So in the 1950s, Rosalind Franklin's using something called X-ray crystallography to determine that DNA is a double helix. She determines that there's got phosphates and sugar on the outside, so like we said on the sides of the ladder, and it's got the nitrogenous bases on the inside. So the rungs of the ladder, like we said before. So basically, she's proven this with X-ray crystallography. This was not known beforehand. Now, Rosalind Franklin was working with a scientist called Maurice Wilkins. Now, Rosalind Franklin was, again, so you could say ethically, she's probably not looking after science the best here as well. She doesn't want to share her information. She wants to keep it really close to her. That's her information. She's going to use it. She's going to come up with a new theory. And that's not how science works. Science works with collaboration and sharing ideas. But regardless, she discovered this. She chose not to share that information. She's working with someone called Maurice Wilkins in the same lab, who's good friends with Watson and Crick. He takes her research and he shares it without her permission to Watson and Crick. They then use that information, refine the model of DNA that they're working on themselves, which they had completely wrong at that point, used her research and they've fixed up what they've done, and they publish their research without ever giving credit to Rosalind Franklin. Okay. Watson and Crick go down, you've discovered the model of DNA, whereas later on Rosalind Franklin then dies from radiation poisoning from the X-ray crystallography she was doing to work out that it's double helix, and she dies in 1958. We then have in 1965, Watson, Crick and Wilkins all received the Nobel Prize for the discovery of DNA, with no credit to Rosalind Franklin. And it's not until 1968, 3 years after the Nobel Prize has been given out, 10 years after Rosalind Franklin dies from the research, and a long longer since she's actually done the research herself and the information has been shared, that she receives credit for her work. So at that point, do we say Watson and Crick wrong for stealing information and publishing it without giving credit? Is Maurice Wilkins wrong for sharing the research? Is Rosalind Franklin wrong for not advancing science and sharing her information willingly? So it's a big debate in science. I'll let you guys think about that. And science is a human endeavour, and we're getting to that next week. We'll be starting that task, but just a little bit of thinking. Your task isn't going to be on this, but just a little bit of information to help you think about those lines. See you in class.